right, so I've placed this is a 1/2 meter by 1/2 meter quadrat here, so 50 centimeter by 50 centimeter. And it's just made out of PVC and has sand in it, so it sinks. And we place our quadrats in our area that we're sampling, and then we measure the depth in the middle. So this is 14 centimeters. We also do an assessment of how many, the percentage of the bottom covered by different things, including rocks and fine sediment, like sand and really fine sediment, as well as organic material, which would be like decomposing leaves or sticks. So I would estimate there's definitely more than half of this quadrat is made of rocks. And there's some areas like here and here where there's a little bit of sand, but not a lot of sand. So I might say about 10% sandy, fine sediment area, a little bit of organic material, maybe about 4%, 5%. So if we said 10% sand, 5% organic material, then the rest would be rock. So that's probably what I would say for this quadrat. So once we assess what the quadrat looks like, then we start sampling in it. To sample the quadrat, the idea is that we want to capture any macroinvertebrates that are in this quadrat. So it's a good idea to keep, this is called a D-net, and it has a 500 micron mesh in there. And so you want to keep this net downstream of wherever you're sampling so that any organisms that get shaken loose go into the net. We will also be trying to get everything into a bin. This rock, if you guys want to look, it's pretty cool. There's a lot of organisms on this rock, including, it's hiding. I don't want to pick up that one. This is I don't know if you can see it. This guy here, this is a helgramite, and they bite. So apparently they feel kind of like a bee sting, so I'm going to be a little careful. I might try to get rid of it just so I don't, I'm not sticking my hands in there with it. Come on. Luckily it rolls up, so I don't know if you can see that. So this is the larva of a what's called a Dobson fly. I've never actually seen the adults. They usually come when they emerge. They're only around for like less than 24 hours as a fly. They have huge wings, but this is near the end of the life cycle in the water. This larva will crawl onto the ground, onto the land, and dig a hole, and then pupate in the ground, and then emerge as this big Dobson fly that emerges at night, so people aren't really that familiar with them. But that's a helgramite larva, and they do bite, so I'm going to put him over there, or her. So when we have a rock like this, you can rub it off in the net, or you can rub it off in the bin. But basically the idea is to get any organisms off the rock. I often have to pick things off. This is probably hard to see from where you are, but this is a little tube of a caddisfly. But basically the idea is to get everything off of the rock that you can. So the idea is to take all of these the rocks that you can pick up and rub them off in the net or in the bin. Here's some freshwater sponge here. There's some other freshwater sponge like right here. This is freshwater sponge as well. And you can see that this one is not green and this one is green. That's because like corals and some other organisms, they have symbiotic algae that live in their cells. So they are green when they have the symbiotic algae. This one was on the bottom of the rock, so it didn't get enough light to have any of the algae. So you can have some that are green and some that are not green. We don't count those in our sampling because it's hard to know what an individual is. But on the data sheet, I would note that there was sponge present in the quadrat. So we can have presence, absence. So The idea is to go through and rub off all the rocks. I'm not going to do all of them right now, but then the idea is once you've rubbed off all the rocks you can move, then at the end you do the macroinvertebrate shuffle, where you shuffle the bottom of the river to get anything off the bottom or in the sandy area, and you must stand upstream of the net so that anything that gets dislodged from the bottom goes into the net. And then you rinse the net off in the bin. Once we've got everything from the quadrat into the bin, we now want to filter the water through this 500 micron mesh little device here. And, sorry, I should have done this before. Take this squirt bottle and just fill it with river water. And make sure to squirt off anything that's in the bin. See a little caddisfly? Yeah. Oh, there's a mayfly. Now you can imagine, sometimes this river is going really fast. It's very slow right now, but there are other times that it's really fast. So these creatures have to be really good at clinging to things, which is good for them, not always good for us. We're trying to get them off of things. Then the last step, now that we've filtered the water out and have just the organisms, there's also sand and things in there, but generally you get the organisms out, you then want to put it in a container. So we take this bag and use our water to squirt everything off. Can you see okay? Squirt everything off into the bag, making sure nothing is left. Sometimes you need to put a little extra water in there. You don't want the organisms to overheat and lose too much oxygen in the water. So we want to make sure there's enough water in there. And then this is called a whirl-pack bag. It's basically got twisty ties at the top. So you roll it over a few times and then twist it. And it's pretty watertight. And then we take this back to lab and sort the samples and identify them there. So in this bin, we have quite a few different organisms. So obviously we have a crayfish. And this crayfish, you can tell is a male by these two little, they're called modified swimmerets, that kind of curve up towards the center. And a female, they don't have those. And they have this kind of triangular area that you can see really well on a female, right in that area instead. So this is a male, it's a pretty big male. You can see I don't know if you can see this, but it's got little orange tips on the claws. It's an Orchonectes virilis, or the virile crayfish. This is the same species, just a younger, hold on a second. This is the same species, but a younger one that you can see also has, ouch. Oh, no. It's just a little pinch, it doesn't really hurt that much. It also has I don't know if you can see this, it's really small, but has the two little modified swimmerets, so it's also a male, and has the orange tips. So this is also a male, but obviously much smaller. And this male, this larger male, could eat that smaller one. And they sometimes will if in a bucket together for too long. So we'll try not to leave them in there too long. Can you focus on this one? I don't want to pick that one up.
Yeah. I'm seeing it, yeah.
Okay. So, well, it's all curled up right now, but this is the helgramite I showed earlier. You might be able to see its gills kind of going in and out. That's what it's using to breathe, and it's got those pinchers on the front. And it might look like it has more than 6 legs or 3 pairs of legs, but most of those things off the sides are really just called lateral filaments, and it only has 3 pairs of legs. So it's an insect.